always hoping that it's going to be primary research literature that gets cited, but not always. This question was almost so basic that a lot of people chose to cite textbooks, for example, instead of research literature, which is okay. The point of the exercise of doing these discussion board questions is to practice finding the research literature, though. So that's what I'm hoping for, but that's not essential. Okay, let's see how we're doing on the quiz. So I got all the possible combinations of 3' and 5' hydroxyl or phosphate. So one question is, what's at each end of the molecule? 5' end is phosphate. So if you have a strand of DNA, the 5' end has a phosphate. The 3' end has a hydroxyl. Okay. So that rules out 2 of the 4 possibilities, and those are the possibilities that had the least support. It looks like 5' hydroxyl doesn't exist, 3' phosphate doesn't exist, so it's either A or D, and D wins. So that's where the new nucleotides are added to the end of a growing strand of DNA is the 3' hydroxyl. In other words, if this really was a piece of DNA, then the next nucleotide would be added there. So DNA grows, we call it, from 5' to 3'. So new nucleotides get added to the right end in this case. Any questions about that? Just something you have to remember. Let's check your other answer. Does DNA replication require the 3' end of a growing strand to start replication? Ooh, about 2 to 1 in favor of true. This is kind of a weird, it's true. DNA replication requires a 3' hydroxyl to start replicating, which is kind of odd. It's a sort of chicken and the egg thing. Like, how is a DNA strand supposed to be, how is synthesis supposed to start at all if you need some DNA there already to start synthesis? And that's one of the topics we're going to get to this class or next time we meet. That's one of the things you should really focus on in the next couple of class meetings is that part of DNA replication. How does that even happen? But it's true, most DNA polymerases, the enzymes that build DNA, have to be able to grab onto a 3' hydroxyl to add a nucleotide onto. And in the course manual, I always provide a section of resources literature that's, I don't remember what I called it, advanced literature or extension. I think I called it extension literature. Those are usually things that if you're interested, if you get bored with what the main content of the topic was, if you want to dig a little bit deeper, go to those resources. In this topic, one of those extension materials, manuscripts, talks about one DNA polymerase that doesn't have this requirement. There's one DNA polymerase from one organism that can make DNA from scratch without starting with an existing piece of DNA. So if you're interested in that, the citation's in the manual. We're still talking DNA replicating, how cells make copies of their chromosomes. Today, and next time, we're going to get into a lot of the vocabulary. We're going to talk a lot about the leading strand, lagging strand, Okazaki fragments, the enzymes that are responsible for DNA replication. And first, I want to give you a little bit of feedback about last class's activities. So last class, one of the things I did was I showed the sequence on the board, and you were all furiously keying out this DNA sequence that I asked you to replicate. And I wasn't very clear with my instructions, because on top here was the original. So this was the sequence I gave. And I got, of all of the submissions, I got 4 that you were doing some extra work, which is legitimate. But what were you doing? What's going on here with these 4 submissions? They weren't just straight up copying the DNA sequence I put.
They were putting in the...
They were replicating. They were putting in the reverse, the complements. C pairs with G, A pairs with T. So those of you that maybe did this and didn't feel like you got very far, it's because you were doing a little bit of extra work, but that's because I wasn't very clear in the fact that I wasn't indeed trying to get you to replicate the DNA to put in the opposite strand. I was just hoping you were going to copy down the sequence that I had shown on the board. Regardless, it looks like these people did a pretty good job of complementing the original sequence, that is, producing the second strand, which is what DNA replication is all about, using a template strand and then producing the second strand that's the complement. C pairs with G, A pairs with T. The rest of you, this is just a subset of all of the submissions. So what I did was, the reason I wanted to collect your writing down the DNA sequence digitally was because I wanted to be able to feed all of that into a computer and ask the computer to do a multiple sequence alignment, like we practiced in class. But computers do well, I don't know. It's up to you, actually. We should discuss if the computer did a better job of this or not. So I took all of the digital information you sent, your copies, and I said, here, computer, take those DNA sequences, and you tell me what you think the best multiple sequence alignment is. So the computer does what we did. It looks for motifs, maybe not so well, and then gives me some sort of an alignment of, so the top line, again, is the original. And so we can compare the original to all of the submissions. And as we discussed last time, the main point of this is where did if there were mistakes, where did they mostly happen? Which we discussed last time, and so I just wanted to show you the evidence that this is usually the case. What were the areas that you had the most trouble, if you had trouble replicating quickly?
Right after the TA.
Yeah, this TA repeat. How many TAs were there? There were 7, I think, if I remember correctly, copies of TA in a row. And that's where a lot of the variation between your responses was. And like I said, this is true. At the enzymatic level, DNA polymerase sometimes itself has problems accurately copying the correct number of small numbers of nucleotide repeats. So this is totally common. This would be something you would see if you actually sequenced chromosomes of humans, not just copied them yourself in some sort of fake digital exercise that I created. Does anybody see any problems with the way the computer aligned these? Computers are not infallible, and they only do what the humans programmed them to do.
The first nucleotide is not the same on each one, even though it's mostly the same for each one, because we all started at the same spot. We all were like, okay, the first one is definitely a C, because we saw that.
Right. So one point is, when there's a hyphen, that means a missing nucleotide. The computer adds gaps if it thinks you skipped copying a letter, for example, put a hyphen in that spot to sort of hold that spot. But in this case, all of this, the original sequence started CACA, and almost all of the submissions got that part right. Some of them did CACT instead. But for some reason, the computer has chosen not to put the CACA as the first 4 nucleotides. It's added 2 gaps there for who knows what reason. Somehow the computer thinks that overall the alignment that it's made is the best possible alignment. Probably not. So there's always a role for humans in hand editing and checking computer output to make sure that actually makes sense. The other thing that's a little bit odd about this is with those TATAs. So for example, circling a few examples where there were TATAs that the computer could have put there where instead it inserted gaps, because that's where the TATA repeat should be. But for some reason over there on the right side, it shoved some of the TATAs off into a different part of the alignment. So computers aren't perfect. And neither are we. So most people got 7 or 8 copies of the TATA repeat. It was pretty good. We will actually use that DNA sequence in the next chapter. So the reason you were copying it down wasn't just for this exercise, but we're also going to use it again when we talk about mutation. Last little bit of feedback before we move on to doing some exercises with DNA replication is the Canvas discussion board with this putative fact, do the 2 strands replicate independently? It was really mixed. It wasn't even close to overwhelmingly one or the other. Hopefully you've all taken a look at some of the comments on the discussion board as well. It looked to me like they basically broke down into some grammar and definition debate, which is great, but this is not a grammar or spelling class. So the questions basically were, does this statement, each strand of DNA replicates independently of other strand, does that mean it can replicate independently, or it absolutely does replicate independently? And really, a bulk of the discussion was about what does independently mean. So we got a lot of questions, or a lot of statements. So here's just an example. I picked a couple of examples from your responses. Statement is not correct because the two individual strands are separated into so forth. Replication fork gets into some of the details. And so separate but similar processes that occur simultaneously. So I think the question here is if two strands are replicating at the same time, at the same place in the chromosome, does that mean they're independent or they're dependent? Anybody have any thoughts about that? Yeah.
The process starts by splicing them into the two halves, so I would think that they don't replicate independently.
So the division of the two strands of DNA, the breaking of the hydrogen bonds, in other words, into two independent single-stranded regions of DNA is necessary for replication to start. So you can't replicate one strand without replicating the other strand at the same time. I'm not sure that that's exactly what the intent of this quote was, which is to say that the conservative method of replication is not what happens. I think that's what this quote, my personal opinion is that's what this quote was trying to suggest, that both strands don't replicate at the same time. They're each replicated by different polymerase enzymes, let's put it that way. And that's kind of what this quote got at. Each strand in the double helix acts as a template for synthesis of a new complementary strand. And they actually cited a quote from this paper: polymerases function independently within a single replosome. So we're talking about, so two strands, this quote from this paper says they do replicate independently. But yes, this still happen at the same time. So, okay, what does independently mean? We'll leave it at that. So hopefully everybody got something out of doing this and at least thinking about how DNA replication works. Now we're going to see the details to whatever extent you want to. A lot of what I'm prepared to cover was information that was in some of the videos that you were supposed to watch for this chapter. I don't want to replicate that to a great extent unless you want me to. Some things were probably not as clear as others. So ultimately, nobody really decided, there was no consensus about whether or not, factually speaking, according to the way that quote was written, replication occurs independently. But the two strands do serve as templates for the second strand synthesis, and that's the important point. The other thing I asked you to do, sorry that I didn't see that this wasn't posted until later after class on Monday, was to sort of draw out DNA replication on sort of a macro scale, not the individual details of what's happening on a template molecule when DNA polymerase is adding new nucleotides, but stepping back and looking at the generation-to-generation, cell-to-cell process of what DNA replication is all about. And so I've got a few screenshots from some of your submissions, just two or three, I think, to fact check, give some feedback. And so I hope you see something that looks reminiscent of what you turned in for this assignment. If you don't, and you want me to give you feedback on exactly what you submitted, I'd be happy to. Please let me know. I'm just going to give a few examples that I think represent a lot of what the submissions were. The really critical thing that I was looking for, though, was what was the color coding here about? Why did I choose this color coding? What do the colors represent? There's blue and black. These are the chromosomal source. Do they come from mom or do they come from dad? So it's hard to see on here. The egg is blue. Maybe it's hard to see. Projector. Meh. The egg DNA double helix in the egg is blue. The sperm is black. So that was the color coding I was used to track where the chromosomes came from at fertilization. The other thing we need to be able to track, though, which I had asked up here, was we need to know which strands are the original strands of DNA that the fetus, the embryo, got at fertilization. We need to know which ones are the original strands and which ones are the newly synthesized strands, and that's why I put that request up there, change the line thickness, not just the line color, because we have to be able to track where the strands of DNA came from. Was it from dad or from mom, black or blue? Plus, we need to know something about, was it an original strand of DNA, which in this example I had drawn as thin lines, and I was asking you, once DNA synthesizes, please draw those as thick lines so we can tell them apart. Most of you didn't do that, that's okay. I just want to look at patterns here for a second. A few examples. So this is supposed to be after replication and division, what happens? So is this example appropriate for DNA replication, and what do you see that you have questions about? Okay.
So after the egg and the sperm, do their thing,
Fuse,
they have a
yep.
full genome
That's
in
correct.
that one cell.
Diploid,
So,
yep.
Yes, the person who put the next two cells, each of the cells has 2 times the amount of DNA it's supposed to have.
Well, I think these are supposed to be base paired, so that would be same, maybe. That's just my interpretation, but let's assume that these are base paired. Let's just assume. But you're right, we have to make sure that each resulting cell is also diploid, has 2 copies of every chromosome. So here it makes it look, from my coloring of blue from sperm, or blue from egg and black from sperm, you don't see sperm-egg hybrid double-stranded molecules. We'll talk about that as we go through the class. But that's the main concern here. What is up with the black and blue hybrid molecules? Does anybody know what that student's getting at? Crossing over. Recombination, which is definitely something that happens, but we haven't gotten there yet. So I'm glad you know that exists. Whoever submitted this, I'm glad you know that this exists, but that was like advanced DNA replication. That's beyond where we're talking right now. The color coding made me a little bit confused on that one. Here we've got the same thing. Every resulting cell is half blue, half black. So it's hard for me to know, without that line thickness, are these all original strands? This is one blue chromosome and one black strand that got together and made a hybrid molecule, or was the color coding being used for tracking which are old strands and which are new strands? Original strands and synthesized strands. So I wasn't really sure what to do with those responses. This one. What's the most important thing about DNA replication? What's the point of cells copying DNA before cell division? What's the purpose of replicating DNA? I'm going to hear from someone in the back. Why does a cell need to copy DNA?
To grow.
To grow. So if a cell divides, the original cell needs to have the DNA. The daughter cell also needs a copy of the genome. So the original cell has to duplicate its DNA so that the mom cell keeps a set of chromosomes, and the daughter cell, the offspring cell, also gets the same set of chromosomes. So the first thing to check here is, do the 2 daughter cells after cell division are they diploid? Do they have 2 copies of every chromosome? Is that what this shows?
Yes.
So we start with, this is a diploid cell. It's got 2 copies of this chromosome. The blue one from dad, the blue one from mom, the black one from dad. Are the daughter cells genetically identical? Do the daughter cells look like they have the same chromosomes? Yeah. They each have 2 copies of this chromosome. So that's fine. In fact, this is going beyond what we've already talked about in class, which is that not only do the daughter cells have to have 2 copies of every chromosome, like the mother cell, they also have to have 1 copy of dad's chromosome and 1 copy of mom's chromosome. So you'd never, in other words, you'd never see the case, or you shouldn't see the case, where you would see one daughter cell get 2 black double helices, the sperm ones, and the other daughter cell getting 2 chromosomes that are both from mom. Wait a second, 1 too many strands. So we shouldn't see those on the right. You always get cells that have one of mom's chromosomes and one of dad's chromosomes. So this is fantastic. The only thing I can't see in this, again, is which were the original strands and which were the strands that were recently synthesized. When I checked yesterday evening, which admittedly was before the deadline, this was the example that I saw that came closest to explaining the synthesis part of this process, which is they created new colors. So we have a blue chromosome here, I think that's blue or bluish, and black. So they got one strand of blue, one strand of black, and then each of those served as a template for yellow, new copied strand of the blue chromosome, and is that green?
Yeah, it's green.
Okay, thanks. New copied strand of the black DNA. So this is the important part about DNA replication is after synthesis, after replication, each parent strand gives rise to a newly synthesized strand that didn't exist in the original cell, the original diploid one-cell embryo. And then I totally understand where you were coming from, student who turned this in. Then at this next generation down here, they gave up on color coding. New copied strands that I was too lazy to make each a different color, which is totally legitimate because the more generations you have, the more colors you'd have to keep track of newly synthesized and not. So that's okay. So this was pretty close to what I was looking at. Two critical concepts. One is that single strands from the parent cell are the templates for new strands that were created by synthesis. So you get hybrid DNA molecules. One strand is from the parent cell. The other strand of each double helix is newly synthesized in that generation. Actually, that is the one most important thing about this. So that's the concept. Double helix separates, and then what we're going to look at the rest of the day is how exactly does that separation happen and how does synthesis start? Does anybody have any questions about this exercise, specific questions about what you might have turned in if I haven't covered something that you want to know about this process? So here's the key to this exercise if I were to create one. The only thing I have to admit is I switched, because of Meselson-Stahl, I switched the thickness. Because remember, Meselson-Stahl used what sort of chemical to label the DNA when they were looking at DNA replication?
Heavy and light nitrogen.
Yeah, so that light nitrogen is normal nitrogen, and then they used a synthetic heavy nitrogen, an isotope that's heavy. In the initial experiment, when they were growing their bacteria, they wanted all the chromosomes, all the DNA to be heavy. So in this case, I switched from the exercise I gave you last time, and I made the thick lines represent heavy. So the thick lines are going to be the original molecules, and then all the thin lines represent DNA that was synthesized after fertilization. So we can have sperm meets black, meets blue, egg, say, it doesn't matter which color is which. So that's the one-cell embryo genotype, just like on the example that I gave you last time. Diploid, 2 double helices of every chromosome, one from mom and one from dad. So then, what is this arrow representing? This is the step we're going to talk about some today. What happens between one cell and one cell stage? What's the arrow doing? What happens to the chromosomes? What does the drawing make it look like the chromosomes are doing? They're separating. What process do we know separates two strands? What can you do to melt DNA, separate the two strands, break all the hydrogen bonds that hold them together? You denature it. So what sort of a condition, chemical or otherwise, could we throw into DNA to make it melt?
Heat or pH.
Okay, so let's go with heat. Yeah, pH probably as well. Disrupts hydrogen bonds. But let's say heat. So do our cells heat up when it's time to replicate our DNA? Is that how our cells denature the chromosomes and get them single-stranded?
No.
No. We've got enzymes in our cells that do that step. But you're absolutely right. That is the process of melting or denaturing the chromosomes. So this is all still in the one cell stage. And then what happens, what's this arrow? You go from 4 template strands. Each of those strands, those are the thick strands. Each of those strands is the template for synthesis of a light, the thin strand. So now you get 4 double helices. So this arrow represents synthesis, or DNA replication. So after DNA replication, then you get cell division. And hopefully then, after cell division, which we haven't talked about, so I'm not going through the details of that necessarily yet. We'll talk about mitosis and meiosis in a couple chapters. Hopefully these 2 cells, after cell division, look identical to the 1 cell stage. So look at where I wrote 1 cell up here. 1 cell. How do they differ? How does that genotype, how does the DNA, what does it look like, and how does it differ from those 2 cells? What are the differences? What are the similarities?
It has half of the thick line and half of the thin line. Is that what you're looking
Yeah, right. So this similarities. 1 cell, just after fertilization, sperm meets egg, chromosomes come together in 1 cell. You've got 1 all blue chromosome and 1 all black chromosome, right? Dad's chromosome black, mom's chromosome blue from the egg. Same here. You've got in each of these individual cells, they're genetically identical to each other, which is what DNA replication and cell division is all about. All of your cells are genetically identical to all of your cells. That's what makes you you. But the difference is absolutely yes, that now this is no longer 2 thick strands. Those are the original 2 blue strands. They've separated into the 2 different cells. There's one thick blue strand, and there's the other one. Yeah.
Shouldn't the last 2 steps, the newly synthesized DNA, all be thick strands and not thin?
So in this case, the thick is the original strands, and everything after that is going to be thin. So all the new strands are thin.
Okay. Thank you.
Yep. Plug that down here. And what I mean by new strands is anything that was synthesized in the organism after fertilization happened. So Meselson-Stahl labeled the parent bacteria with heavy nitrogen. Then he got rid of the heavy nitrogen and the bacteria started dividing, so that all the DNA that the bacteria made as they were doing this experiment, they were making light strands, normal DNA strands. So then after we do the next 2 steps, which are another melting step, denature the DNA, and then another synthesis step. This is 1 on the bottom left. This is 1 of 4. Well, this is just before cell division. What's the difference between this cell up here, right before cell division, and this cell in the 2nd generation, just before cell division? This is the critical difference. This is what Meselson-Stahl saw in their experiment, which we look at next. Each of these strands has, or each of these double helices has an original strand. They're each one of those is half heavy, half light. 1 thick, 1 thin strand. Down here, after the 2nd cell division, or just before the 2nd cell division, there's 2 heavy strands out of the 8. So you have 2 half heavy strands and 2 entirely light strands. That's the only outcome you would predict if semi-conservative replication was happening. Every generation, if you take any notes, this is the one. Those of you that have been sleeping in the back and browsing YouTube. Every cell division, you get more all light or all brand new strands compared to the original cell. In other words, when you were born, there was 1 sperm that met 1 egg. Every one of your chromosomes, when you were in 1 cell stage, which every one of you was, even if you were a twin, even if your twin's not enrolled in the class, every one of you started as a 1 cell embryo. Every one of your chromosomes, you had 4 strands. 2 strands, 1 double helix from your dad, and 2 strands or 1 double helix from your mom. You never get more original strands of DNA. Those are the ones you were born with, the 1 cell stage. Everything since you were born is newly synthesized while you were an independent organism. So somewhere in your bodies, presumably, you still have those original 4 strands left. Maybe. It's possible. Unless they got destroyed somehow. You initiated from 4 strands of DNA for every chromosome, the 1 cell stage. Everything since then is thin, the light, the newly synthesized since you were created, conceived. The whole point here is that the more generations you go, the more dilute, the more rare those original strands are. And that's what Meselson-Stahl we're looking at. Generation after generation, what happens to those original strands of DNA? So to really explicitly tie this to what their experiment saw, I'll remind you that this is the original figure from their paper, and they're looking at measurements of, are we looking at chromosomes, double helices, that are made up of both strands that are heavy, both strands that are thick chromosomes. That would be a band at the bottom of the microfuge tube, which corresponds to that band on this image. So that's all heavy. And that's like what they see in what we saw in that key at the original generation. All the strands of DNA are thick. Those are the heaviest, the densest DNA molecules. They went the farthest down the centrifuge tube. So that's what that cartoon corresponds to. Every single strand of DNA is heavy. So that's the bottom or the rightmost, the way they took the picture, bands. And then by the first generation, Meselson-Stahl saw this band. There's only one band in all the bacteria at the first generation of growth. And what's true of all of the chromosomes in an organism at the first generation? They're not all heavy anymore. That's this size band. They've moved a little bit left or up the tube. They're a little bit lighter in weight. So those bands, let's color them red, for example. They're a little bit lighter. Those are the half heavy, half light cells. Every double helix has a heavy original band and one newly synthesized light band. So when they're double base paired, when it's double-stranded double helix, those are not quite on average as heavy as the original. Both strands are thick. 2 thick strands heavier than 1 thick, 1 thin. So what happened at the second generation? If you go down that number, I know they didn't measure even generations, 0, 0.3, 0.7, 1, and then 1. I don't know how they calculated generations. I don't know how you do decimal generations. But when you get close to 2, 1.9, what band sizes do they see now? What's that band size? That represents the half and half. Same thing all the way down there. So they've got a half heavy, half light band there. Half H, half L. And then what's the new band that's shown up for the first time? This band. That's all light. Those are the first time in generation 2, the first time you get synthesis of all light strands, which is what we already saw. Right. There and there. After this next cell division, you, for the first time, can generate strands, both strands, all newly synthesized since conception or since birth. So if you look down time, last point for this figure, this is time equals 0, basically. 0 generations. Here we get to 4 generations by the bottom of the figure. So we're watching time progress as you look from top to bottom on this figure. What bands disappear and what bands show up? There are 3 different bands. We've got heavy, we've got half heavy, and I should draw in light too, just for fun. So what DNA, what type of DNA? Heavy, heavy, heavy light, or light, light, disappears over time. We start here, I look down. What goes away? This all heavy. Why is that? Because the only time that the two original strands are together is the first generation. And then every additional generation, they get separated into different double helices until you can't even see them by the first generation. There's no more thick left. By the first generation, it's all the original molecules, the thick, have been used as templates for synthesis of a new strand. So by the first generation, you totally get rid of all the original DNA, double helices. And so then as time progresses, the half-heavy, half-light bands get fewer and fewer and fewer for the same reason. You're a big body now, you've got lots of cells, but you only have 2 copies, 4 strands, of the original heavy DNA. So they kind of get diluted out. And then by the second generation, for the first time, you start seeing all light, all newly synthesized. And of course, that just keeps getting darker and darker, because by the time, if we were talking about bacteria, it's different. If we're talking about humans, by the time you get to be 1 trillion cells, in your 1 trillion cells, you've only got 4 of those original double helices, or 4 of the original strands, 2 double helices. So all of the DNA in your body by this point has been synthesized fresh since you were conceived. That's the Meselson-Stahl experiment. It's totally incompatible with any other form of DNA synthesis, which is what we talked about last class, which was conservative replication. You wouldn't see this pattern of bands disappearing and appearing if your whole double helix at one time was just copied and made a new double helix. And I might even ask you to predict what sort of pattern you would see on this sort of an experiment if conservative replication was what happened instead of semi-conservative. Hint, hint. I'm not recording this either, so hopefully you were paying attention. Kidding. It's in the... Go back and listen, because that's something I might ask you on an exam. So now's the point in time when I have to admit that I have falsified, or at least I've made an oversimplification. This is not how DNA replication works. So I asked, how do you melt the strands, temperature, pH, enzyme? How long are our chromosomes, base pairs wise? Thousands, millions, billions, trillions?
Billions?
Hundreds of millions, tens of millions. They're long in terms of numbers of nucleotides. So it turns out that linear chromosomes, like humans have, like straight chromosomes, bacteria have circular chromosomes. We have straight chromosomes, linear chromosomes, that have 2 ends. Our chromosomes do not do this, which is what I used in the cartoons. They don't all separate the whole length of the chromosome all at once. So your chromosomes will not look like this during synthesis. They do something else. So what I'd like you to do, please, and we're just going to do a quick practice, and then we're going to close. Google Classroom. You should have seen an assignment show up at the start of class, but it should be sitting there waiting for you. Just imagine that this is part of a huge, long, linear chromosome. That's what the dashed lines on the edges indicate. More DNA goes in either direction, but this is the spot in the chromosome where replication is going to start. What do you think this double-stranded double helix looks like just as soon as replication starts? Draw me what you think. You don't need to know. I just want to know. Draw what you think happens underneath this double helix. What's this going to look like a couple minutes, say, after replication starts? So take 2 or 3 minutes to do this. One of the reasons to do this is, again, to practice PDF annotation, send it back to me in preparation for exams. The other reason is I'd like to know how you feel about this before we go on. And if you have a question, please let me know. So this is not meant to be perfect. Do it quickly. Get the gist of what you think is happening. You don't even have to draw the nucleotides if you don't want to. Just a drawing of the strands would be fine. All right. So let's see what we've got here. So here's one. It's a little bit light, but that's okay. We've got the double helix here and keyword, unzipping, which you can kind of think of double helix as a zipper. It has to be unzipped. There's a weird, we'll talk about it later. Yeah. Something about your genes being unzipped. It's probably in poor taste. Google it if you want to. There's some sort of meme about it. So the DNA double helix has to be melted. In this case, this individual that turned this in is indicating that right here, it looks like, is where the unzipping, the melting of the double helix is starting. So what do we call that? The place where the double helix just starts to melt, denature, is the origin of replication. So in other words, if you want to know where a chromosome is going to start replicating, you look at the very first spot where the two double helices separate. That's where replication starts. Let's take a look at one more. Okay, same idea, just a different side of this part of the double helix. You've got the double helix separating, and then the replication fork, which is right there. It looks like the double helix splits into two. It kind of looks like a fork, I guess. The replication fork is the position where the two strands are separating. So the origin is the very first spot in the chromosome where the two strands come apart. But the fork then moves, as this student is describing, unzipping the DNA as it goes. From the origin of replication, the most critical part here is it just going in this direction? Because there's more chromosome over here. So there are actually two replication forks. The other direction from this origin of replication would also be unzipping. So you start in the middle of the chromosome, and you replicate in both directions from the origin. So that's the take-home message from this assignment. In other words, if you draw both of the forks at once, there's our initial state, and then at some point you're going to see something that looks like that, which is called a replication oval? Bubble. Replication bubble. And that's the replication forks, the DNA unwinding in both directions, the black arrows, at the same time. And then you've got nucleotides hanging out here, not hydrogen bonded with each other, and that's where DNA replication starts. Now the polymerase, the enzyme, can grab onto the single-stranded DNA, and it can read, sort of, in anthropomorphic terms. It can tell, for example, that that's an A in the bottom left, and that it should bring in and add a T to complement it. And that's the beginning of the synthesis of the second strand, which we'll talk about more now. So a couple notes about next time. Please go to Socrative now. We've got a couple minutes left. I'm asking you one question on Socrative, just feedback on what you think you learned today, what you'd like to learn more about. Friday, I can't be here with you. So I'm asking you still to do some work. Just don't bother coming here unless you want to. If you want to work together on this or meet in the union or wherever, that's fine. I'm going to post replication bubble exercise, like we just looked at, that kind of structure. I'm gonna ask you to label some things and add some details. That'll be a PDF that goes to Google Classroom that I'd like you to work on Friday during class time, and then send that back to me by next class, that is on Monday. And then other things to do for Monday. Two other things. Monday's the last time we're gonna talk, I think, we'll see, about DNA replication, so it's a good time to do the last fail exercise in this chapter, which leads us into mutations for the next chapter. The last thing, most importantly, one of the extension materials in this chapter is this paper, Moeller and Niedzinski, 2012. I'd like you to find that paper, and I'm going to leave a few instructions on a couple of paragraphs, not the whole thing, I don't want you to read the whole thing. Read the paragraphs that I suggest you read, and look at the figure that I suggest you look at, and I want to be able to have a discussion about just that one figure from this paper on Monday as well. It is all about replication origins, and that's where we're going to head on Monday. So don't come Friday, work on these things Friday and over the weekend, come prepared on Monday. And I can hang out for a couple minutes and answer questions if you have any, otherwise I'll see you on Monday.